My name is John Carpton. I'm chair for the Department of Translational Genomics, the USC Keck School of Medicine. And today I'm going to talk a little bit about tumor heterogeneity. And the sort of the structure of this talk will be, I wanted to walk through an example of why this is important, why it's important. We've seen many papers from Charlie Swanton and others, but I want to walk through another live fire exercise that was the result of a clinical sequencing project where complications in copy number detection actually led to, sort of an interesting story on why we really need to understand heterogeneity in precision medicine. And so I'm going to really focus on this, and I just want to thank Anushka and Edwin for the opportunity to present today on 10x Genomics' new tech and the promise of this technology. So many of us have seen and read the literature, the work from Charlie Swanton's group, where they took a single renal tumor, broke it into various pieces, and then sequenced those regions independently, where they selected different regions from a renal tumor and performed next generation sequencing. And we're actually able to provide some context around which regions were likely to give rise to the metastatic tumors from that same patient. This particular region, R4, contained mutations in common with the metastatic lesions, but none of the other primary regions did. And they were actually able to use that information to develop phylogenetic trees on the likely pathogenesis of this tumor through its course and progression. And so I want to sort of talk about some work that we've been doing in studying tumor heterogeneity, but in the context of precision medicine, where the information was used to actually identify therapeutically actionable events to treat patients. This paper was published in collaboration with our partners at the Mayo Clinic in Scottsdale, where we identified therapeutically relevant context around alterations in FGFR2 and the ERB receptor pathway in sporadic intrahepatic cholangiocarcinomas, and actually treated patients based on the results. Okay, as the most frequent form of biliary malignancy, it can be diagnosed in either a sporadic form or as a result of liver fluke infections, particularly in Southeast Asia. It can be very difficult to treat, typically treated with surgery, chemotherapy, radiation, and palliative care. Again, these tumors tend to occur within the bile duct system and can be intrahepatic, perihilar, or distal extrahepatic. In collaboration with Alan Bryce and Mitesh Borad, as part of a clinical sequencing program we had in collaboration again with Mayo Clinic, we had a patient that came in with liver biopsies that showed moderately differentiated adenocarcinoma in the liver, which was later diagnosed as advanced cholangiocarcinoma. We were able to accrue the patient onto the protocol, collect specimens for genomic analysis in a clinical setting. So just a depiction of the copy number plot for this particular patient. Seeing regions of loss and gain, we pointed our attention to this region on chromosome 8, which was a high-level amplification and a relatively complex rearrangement. And after further interrogation, we also identified mutations in ERFI1, which is the endogenous inhibitor of ERB receptors. So complete loss of ERFI1, which maps to 1P. So we had the deleted indel in the coding region as well as LOH, and a mutation in the P53 gene. Further analysis of the region on chromosome 8 revealed an oncogenic fusion that included NRG1, which is a ligand of ERB receptor, specifically ERB2, and oncogenic fusions that include NRG1 have been detected in other cancers and are known to activate the HER2, HER3 axis. Again, we also identified these coding mutations in these two genes as well. So this particular tumor, from our perspective, had a double whammy. We had sort of oncogenic activation through high-level ERG1 fusion oncogenic expression, and we also lost the brakes. So ERFI1 encodes MYC6, so we had biallelic inactivation of MYC6, so we sort of had dual activation of this pathway. Further discussions during the molecular tumor board and further knowledge mining suggested that we would see activation through ERB2 and ERB3. Thus, we selected the use of pertuzumab, which inhibits ERB receptor heterodimerization. And we actually put the patient on treatment and saw an incredible response with about 2 or 3 cycles of pertuzumab. Patient did respond, but we saw a relapse, of course, about 5-6 months later. We were actually able to obtain a second biopsy from this patient. And interestingly, upon profiling this tumor, we saw the almost loss of the 8P alteration, but this high level amplification of EGFR that wasn't there in the original tumor. Right? I mean, there was no indication of this high-level EGFR amplification, but still, the cholangio being driven by this ERB receptor activation axis. Interestingly, we also saw the ERFI1 mutation and the P53, which suggested that these two tumors were clonal. So we believe we had 2 independent tumors growing in this particular patient, one being driven oncogenically through activation of NRG1, and a different clone being driven through activation through EGFR. Patient went on erlotinib, and there was a response to erlotinib. Of course, the cancer came back. We're actually able to get another biopsy, and guess what happens? The EGFR event goes away, and the NRG comes back. So we had this sort of clonal tide occurring under pertuzumab versus erlotinib. And so this is just an example of why it's important to be able to identify these clones early and not have to wait after 2 or 3 rounds of treatment, because the problem here is when the cancer comes back after erlotinib, most cases an oncologist isn't going to go back to the original therapy because that patient has already failed that therapy. Right? So if we can identify this early and know that there are multiple clones, perhaps we can take a different approach to treatment through sequencing of drugs or through combinations. So being able to decipher copy number and ploidy changes in mixed cellular populations is more difficult because the signals can get diluted. We can identify mutations in multiple clones, but in many times with copy number, the signals get diluted with these multiple clones. And so outside of FISH, single cell analysis holds the most promise for identifying these clonal populations that contain drivers that can be altered by copy number changes, as in the example I gave with cholangio. Current single cell DNA methods rely on highly laborious brute force techniques. Thus there exists the need for novel approaches to generate genome-wide sequencing libraries from single cells. Enter 10x Genomics Novel Tech, and this is work that was done in collaboration with 10x and Claudia, Shomani, Edwin, and the team, and at USC, Enrique Velezquez, David Craig led on the analytical side, and myself and Lee Gibbs were working with Shomani and the team on some of the and Claudia on the wet lab aspects of this. And so Anushka talked a bit about the technology. Essentially, it's combining beads through emulsion, cell beads with gel beads. The gel beads contain the reagents for sequencing along with unique barcodes, so that every sequencing library that's generated from a single cell has a unique barcode. And so this is beautiful because now we can map the reads back to single clones. Then the sequencing reads are then generated on short read sequencing. The beauty of this also is that we generate true ploidy. So it's not looking at normal versus tumor and generating log fold changes like CGH. We get actual true ploidy information as was described by Anushka in her presentation. And so in a small pilot study, we've run 4 real tumor samples through the system. They all happen to be renal cell carcinoma samples. We had 1 stage 3, 3 stage 1 or early cancers, 2 from white patients, 2 from African American patients. And we generated data on roughly 1,000 cells for each of these tumor specimens. And these were essentially fresh specimens where the single cells were dissociated using mechanical methodologies. So this just depicts a distribution of reads per megabase, looking at the number of reads per megabase on average for the 4 samples. And here are the heat maps for these 4 samples. The stage 3 cancer showed a highly aneuploid a picture of its copy number and ploidy. Importantly, we had one tumor that only had 51 tumor cells, and from that we were still able to deduce clonal heterogeneity, which to me is an incredibly powerful aspect of this technology. Just walking through these as well, looking at the median ploidy for the 4 samples. Again, one of the samples with an incredible degree of aneuploidy, and 3 that looked a little bit more normal, as we would in terms of the stage 1 cancers. So this is, again, the depiction of the stage 3 cancer, and we can see this incredible amount of aneuploidy. And focusing just on the tumor cells, we can see a series of clonal populations that are based on very distinct copy number alterations that are very specific for these specific clonal populations. This is about 267 cells. Here's another this is a stage 1 tumor, and that had a relatively stable, relatively standard renal cell copy number profile with alterations occurring on odd number chromosomes 3, 7, and 13, and 17. Again, a zoom in also shows that there was some clonal distribution in this, but this was a relatively monoclonal tumor from our perspective. Again, this was one of the more interesting samples, again, in that most of the cells, 820 of the cells, happened to be diploid, but we saw a small population of cells, about 51, that actually had alterations indicative of renal cell carcinoma, including loss on chromosome 1, 3, 9, and 10. And again, zooming in, we can still see, even with 51 cells, we can still see nice clonal structure and cellular heterogeneity in this particular tumor. Now, the other great thing about this technology is that because the cells are individually barcoded, we can then go back and look at the copy number plots of individual cells. And to be able to deduce this level of heterogeneity, from my perspective, is incredibly powerful. Now, of course, we've gone beyond the current specs